and welcome to a new episode of the Computomics Podcast. Our guest today is an expert in plant molecular biology and biochemistry and is the managing director of the Leibniz Institute of Plant Biochemistry in Halle. Welcome, Alain Tissier.
Thank you for inviting me.
It's our pleasure. Alain, a little Computomics birdie told me that your second biggest passion is music and that you play the oboe. What is your favorite piece of music you play with the oboe and why?
That's a tough question.
We like to start with those.
Yeah, there's a lot of Baroque music from Bach, Handel, Vivaldi, and so on. But there's one big piece of music from Mozart, which is one of my favorite pieces for wind instruments. It's called the Grand Partita. It's a serenade for wind instruments, and that's a beautiful piece of music that I really enjoy playing.
I will have to listen to it maybe later today. Mm-hmm. Thank you for sharing that. You're obviously one of your other passions, maybe your first passion, I'm going to assume, is biochemistry, plant biochemistry. I was wondering, after your postdoc, you worked at an atomic energy research center in France. How did that influence your work in plant biochemistry?
So I did my PhD and a postdoc. PhD was on DNA recombination, and then a postdoc on using transposons to create mutants in plants. And so I was still in the continuation of this work. I was interested in DNA repair, DNA recombination. So it was a logical step in my position in this center, because I worked on the effect of radiation on plants, how plants react to radiation. And one major effect of radiation is on DNA, on damaging DNA. Mm-hmm. So that was, of course, like I said, so a logical step from my previous work. So there, working on DNA repair, I was also interested in rearrangements in the genome, in particular between repeated sequences. Mm-hmm. And then in those days, it was the time when plant genome sequences were first published. So it was actually the Arabidopsis genome, the model plant Arabidopsis thaliana, that was sequenced. It was published in 2000. And we had access before that already to aspects of the genome sequence. And what occurred to me is that there was a lot of repeats in the genome of repeated genes, clusters of repeated genes. So I was intrigued in this. And this led me to look at what kind of genes were present in those repeats. And there were a lot of genes involved in metabolism. So that's how I got more involved into metabolism and what plants do with those genes that are repeated.
Interesting. The natural follow-up is, what do they do? So basically, I think you focus on the interplay between the primary and the secondary metabolism, right?
Yeah, that's one aspect. But actually, these clusters of repeated genes, they are often, not only, but often involved in the production of metabolites, of small molecules that contribute to the defense of plants against various organisms, so pathogens, disease-causing organisms, but also to interact with other organisms, to communicate with other organisms, with insects or with microorganisms. And so those repeated genes, they are basically a reservoir for evolution of new functions. And this is important to create also this chemical diversity that plants can produce. Plants are really master chemists. And through these repetitions of genes that can evolve then independently and create new functions, that way you create also chemical diversity.
Interesting. I think in one interview, you refer to them as little chemical plants, this part of the plant biology.
Yeah. So, and often also they produce those chemicals in specific organs, in specific locations that are really dedicated to the production of those metabolites, those molecules. So those organs in the plant are kind of mini plants in the sense of industry plants, to produce those compounds. In particular, those secretory organs like trichomes, these are the ones we're particularly interested in at the surface of leaves, but you also have glands or resin canals, for example, in pine trees, if you cut a pine tree, they exude this resin. These are specialized structures that secrete those metabolites in really large quantities. So these are the kind of things that we're interested in. And there, because they're able to produce those compounds in such large quantities due to those specialized structures, is then the connection to primary metabolism. How do they achieve to supply those structures with enough material, let's say, to produce those toxic compounds, often toxic compounds in such large quantities?
Mm-hmm. And I assume that's quite a wide field that covers a lot of specific aspects. At the IPB, the Leibniz Institute of Plant Biochemistry, what would you say are your main areas of focus within that wide field?
So one really unique feature of the IPB, it's a plant biochemistry, but we have a chemistry department. And with the chemistry department, we collaborate so they can, for example, synthesize compounds that we identified in plants or in other organisms. We can elucidate the structure of these compounds. And so there's a really tight collaboration with the chemistry department. They also look in the chemistry department for new bioactive compounds from plants and microorganisms. But other than that, the IPB, I would say, is interested in how plants react to their environment, whether it's with, for example, with pathogens or disease-causing microorganisms, or with abiotic stresses. So, for example, nutrient supply or temperature stress. So basically how plants were interested in understanding how plants react to their environment at the biochemical level. So what are the metabolites that are produced that are contributing to these responses, but also how they are produced, the transduction signal pathways that lead to the production of these metabolites, for example.
And your research specifically, what do you focus on?
So in my group, there are three main topics. One is to understand these cell factories that produce those metabolites, so specifically glandular trichomes. That's one aspect. One aspect is also we worked on a class of compounds that are called terpenoids. Terpenoids, you have, for example, menthol, from mint is a terpenoid. There are many compounds, aromatic compounds that are terpenoids. It's a very important class of compounds. So we work on the biosynthesis of these compounds and how we can engineer and create more diversity by combining enzymes involved in the biosynthesis of these compounds. This is what we call combinatorial biosynthesis, and with the idea of identifying new bioactive molecules. So, exploring the chemical space for new bioactive molecules. And then another aspect is engineering. So we do metabolic engineering in plants and yeast, for example, but also genome engineering. So genome editing to particularly with a new technology that we developed to insert at a specific location in plant genomes sequences. And this is the basis of our INNO-TOM project that is just starting now.
Let's jump into the INNO-TOM project with a little more detail, maybe. You just mentioned a specific genome editing technology that you've developed and are using in the project. Can you explain what that is?
So, as you probably know, CRISPR-Cas was discovered about more than 10 years ago. And this has really revolutionized molecular biology. And in plants in particular, it gives you the possibility to introduce breaks or to modify DNA at specific positions. But in plants, if you want to insert a sequence or modify a sequence in a targeted way, not just making a cut and seeing what happens, introducing a kind of random mutations, but if you really want to introduce at a specific location another DNA sequence, for example, this is much more challenging because the way the plant does repair those lesions that are introduced by CRISPR-Cas is called non-homologous end joining. So it's repairs, it brings the two ends together and just repairs it that way. So the technology we've developed is based on CRISPR-Cas, but we added another function to it, which is an exonuclease, a 5' exonuclease to be precise. And what this does is that when the DNA is cut by CRISPR-Cas9, for example, it will digest one of the two strands of DNA to create a free single strand, which can then be used to repair the DNA on a template DNA that is provided also together with this old mix. Yeah. So it's basically you provide a template to repair the DNA, and you generate the substrate in the genomic DNA that is more easily repaired that way.
And what are the advantages of that? So does it reduce lesions? Does it make it cleaner? Like, how would you describe?
It makes it cleaner, and you can also insert at the position where CRISPR-Cas cut the DNA, you can insert any sequence that you want. Yeah. So, for example, if you want to modify a gene sequence, so that it acquires a new function, then you can do it this way. Or if you want to modify the promoter of a gene, the promoter is the region that defines when a gene is expressed or not, then you can alter the pattern of expression of this gene. So you can make it express in a different tissue or responding to a different stimulus in the environment, for example. So it gives you a lot of flexibility, a lot of opportunities to alter genes in a way that is much more precise and specific. And also you can use sequences that you insert in the genome that come from the own genome of the plant. So that's what we call cisgenesis. So meaning you don't use transgenic material to do this, or it's actually an option that you have. You can, but you have the option of using the plant's own material to modify its genes.
So intragenic modification, basically.
You can call it like this, yes.
Interesting. And what plant are you working on for this project, and why?
So in INNO-TOM, we're working on tomato. And one of the goals of the project is to modify a gene for resistance to a virus. So in plant breeding, you often cross related wild species into the cultivated species. Why? Because those related wild species, for example, have genes that confer resistance to pathogens that the cultivated species does not have. They don't have those resistant genes, or they have copies of those that do not work so well. Let's put it this way. And then what you can do is take those genes from the related wild species. They are similar but different to copies of the genes in the cultivated genome. And then using our technology, then you can replace them with the copy from the wild genome. So you can do this with traditional breeding, but it takes much longer. And also the problem with traditional breeding, particularly when you cross with wild species, is that you can have large regions of the genome that you cannot recombine because they have rearrangements. They look a little bit different from the cultivated genome, so that it's difficult to break it down to small pieces, to only the piece of genome that you want. And with our technology, we can do that precisely. That's one advantage. One goal.
So you said precision and speed are like the major.
Yes, exactly. And another thing we want to do is I don't know if you've heard of this, there are purple tomatoes that have been developed by a group in the UK, the group of Cathy Martin. They are transgenic tomatoes. She introduced two genes that make the tomato purple. They're purple because they produce some compounds called anthocyanins that give the color, but are also good for your health. But you could do this using the tomato's own genes, but you need to modify them. And that's what we want to do using our technology. And then that way we can have those purple tomatoes, but non-transgenic, let's say.
Interesting. That sounds like it's pretty revolutionary work that you're doing. How far along in the project are you, or what's the timeline we're looking at?
So the project is for 4 years, the first part of the project. So hopefully by the end of these 4 years, we will have, let's say, prototype that we have generated in tomato. And there's possibly a second period of funding that could be possible. And we have on board of this project, we have two breeding companies, KWS from Germany and Richtwan, it's a Dutch company, but they're also involved in Germany. And so the idea is that we transfer the results of this project to these companies so that they can develop them, commercialize them.
I think that's actually something to emphasize in this project. Like, what is the partner structure, or kind of the overall approach isn't just to look at, okay, how can we modify the genetics of this plant, right? At the research level, you have a much broader scope. Can you describe that a little bit, please?
So in total, we have 5 partners. Actually, what I described is mostly what we will do in my group. There's also Professor Thomas Lahaye from the University of Tübingen. And he will work on bacterial disease resistance, also in tomato. And we have Computomics, of course, the bioinformatics company. They will assist us in identifying cis elements, so elements in the genome that confer, for example, expression in the fruit, but also to identify disease resistance genes, for example, bacterial disease resistant genes. And we have those 2 plant breeding companies, so KWS and Reiswan. Both companies have part of their activities is also on tomatoes, so Reiswan in particular, but KWS also for a number of years now has been investing in vegetable breeding, so tomato breeding. So that's the structure of the group. And the idea is also to use tomato as a kind of model, a proof of concept to show what we can do with these technologies. So combining genomic sequence information with new editing technologies to create new breeding concepts to introduce beneficial traits in plants. So either health beneficial or disease resistance.
Mm-hmm. And is there an estimate how that will impact the speed to market? Because you emphasized precision and speed so much earlier. Like, will it help, for example, addressing climate change issues where maybe you need plants that are heat resistant much faster than normal time to market will allow for?
Yeah. I mean, climate change, of course, there's the climate aspects, but also there's other consequences of climate change that you have, for example, pathogens. You will have new pathogens that will become, that can then develop in colder regions. You have spread of pathogens that are not, for example, in Europe yet, but will come in Europe soon or are already beginning to emerge in Europe. So that means you need to react also faster to be able to breed disease resistance traits faster, because you have to be able to respond to these changes that kind of occur very fast. So the ability to introduce, for example, disease-resistant genes at a specific location, and also I would say, in a non-transgenic way or cisgenic way, then this would allow, first of all, to speed up the breeding process, but also, since hopefully in Europe, you won't have to deregulate because they're not transgenic, to deregulate them also. This creates opportunities to bring them faster to the market and also to make them more acceptable. But in terms of pure breeding, I would say if you do traditional breeding, you will need at least 10 years, to introduce a specific trait into your, let's say, elite cultivar. And now with this technology, I would say you can reduce probably by half, at least by half, the breeding time. So I think this is, it really provides opportunities to speed up and therefore to react faster to, emerging diseases, or also, as you were saying, to breed plants that are more resistant to, specific climates, or whether it's heat or, other climate events. So, of course, these are more complicated. So I would say disease resistance is a more simple one, because typically you have one disease resistant gene, a single gene that can confer resistance to a particular pathogen. But for more complex traits like resistance to drought or to heat, this may take longer, because it's a multifactorial trait. These are multifactorial traits. So this is more complex and will take more time.
But overall, and maybe as a final question, how confident are you that your research will yield results that can be put into practice beyond this research project, that can be applied to in other scenarios?
Of course, it's research at this stage. You never know, but I think it's important, and we're quite confident that we can deliver some important results. But as I said, it's research, so you have to be always cautious, not promise, the solve all problems. You have to be cautious and not over optimistic. But I think really CRISPR-Cas, and not just the technology we have developed, but also many other aspects around CRISPR-Cas can really make a difference. In the end, we have to see what traits really make it through, really bring advantages. But this definitely creates a lot of opportunities. And to be able to breed plants that adapt to new conditions, climate change. This is, of course, a major issue. Increasing yields. This is still a major issue. So there's a lot of challenges. I think agriculture is going to be the major challenge of the 21st century. And so that's why we need a lot of research and technology development in this area. I think is very important.
100%. Alain, thank you so much for spending some time with us today and giving us insight into your work, especially the INNO-TOM project. It sounds amazing. We will link to the project site on the show notes to make sure, if people want to check out more details. Are there any other places that you would recommend that we check out?
I think there's another interesting site just regarding plant gene editing. It's called SAGE. It's a database of gene editing events in plants that are produced through research, or also that are commercialized now. There are some that are being commercialized. So that's for people who want to get an idea of what's happening in this field. I think this is an interesting place to look for.
Great, thank you. We will definitely link to that as well. Other than that, I hope to have you back maybe towards the end of the project at the latest, so you can report on your findings. For now, thank you and talk soon.
Thank you very much, and with pleasure.
Thank you for listening in today. Want to learn more? Then check out our show notes and more info on Computomics.com. If you have questions or want to propose a next guest, please reach out to us at podcast@Computomics.com.